slide, right? Interphase is the resting state. Interphase, there are things happening, but this is a normal cell. So in your lab manual, you have circles to draw. Yes, no? Can I erase this? You can write it down. I'll see what I have. Interphase has 3 parts to it, though. You have to know the 3 parts. So on page 73, it says interphase, and it says G1, S, and G2. Do you all see that? So you're going to write the following. You ready? Now I know this is interphase because it has an intact nucleus, right? Cell membrane. All this stuff is called chromatin. You need to know that. All the genetic material in here is not very distinct. It's called chromatin. But let's talk about G1, S, and G2. And I'm going to simplify this. First of all, know that interphase basically is the resting stage. The cell is not actively dividing. Some of your cells never leave interphase. Some of them are stuck in interphase because they don't divide. They don't replicate. Tell me a cell in your body that never leaves interphase. Will never divide. Nerves, a lot of your nerve cells, not all of them, but your gray matter in your brain and spinal cord. That's why if you injure that type of tissue, are you in trouble?
Yeah.
It's non-repairable. Cartilage is another one. So, G1, the cell grows. Anytime you see a G, you should think growth. The cell has just divided, and so it needs to grow. Now, also in your lab manuals, if you look on page 68, there's a cute, nice little chart there showing you. Do you guys have lab manuals?
I don't. I'm gonna get this right.
Do you have your Connect card still works? Or no, it wouldn't work.
I bought it online.
Okay, so you're getting a little lab manual too online?
No, I'll just buy it.
You can get that.
Yeah, I will.
All right. Anyway, there's a nice figure 7.1 that shows you in color, and it tells you G1, the cell will grow. Because if it divides, it is half the normal size. Yes, no?
Yeah.
It splits in 2, so it has to grow. S part is for DNA replication. Why would your DNA need to replicate?
Because it only has half of the DNA it had before.
What?
Because it only has half of the DNA.
Can't hear.
Because it only has half of the DNA
No,
it had before.
that's not right. It got the full DNA. If it didn't get the full DNA, it couldn't be a cell copy.
Because it's getting ready.
It's getting ready in case it needs to divide again. And this is why they're hopeful that even in a spinal cord injury, if we could somehow get that cell to actively divide, the DNA should be replicated already. Should be. In case, because your cells need to divide it quickly. So that DNA takes a while to replicate itself. Okay, G2 is when the cell completes growth. So anyway, if you see the G's, you should think growth, right? But otherwise, interphase is just a normal cell. And now this cell, which looks like a fried egg always to me, what's this part here?
Plasma membrane.
Plasma membrane. What's this structure here?
Nucleus.
Nucleus. What's all this stuff?
Chromatin.
Chromatin. This is actually a central, but it's hard to see. Does this look different?
Yes.
Okay. Now you start to see, first of all, what is gone. And guys, a lot of things happen in prophase. And I'm going to start to name what happens in prophase. And you should write it in that little box on page 73. So hopefully you'll remember it. I'll write it somewhere. All right, so number one, prophase, nuclear membrane fades. It's actually reabsorbed here. We gotta get rid of it. Otherwise the cell can't divide. The chromosomes thicken, shorten, and they double. That's really important that they double. They have to have a copy of themselves for the new cell. When they double, you can see them, and those double chromosomes are now called chromatids. Think D for double. So all these black squigglies are chromatids. Can anyone tell me, if this was a human skin cell, how many chromosomes would be in the cell right now?
46.
46 is incorrect.
It would be.
So 90.
Oh, because it doubled.
Doubled, so it'd be
46.
92. Couple other things happen. Number one, the centrioles move apart. Centrioles move apart and start to form spindle fibers. So the centrioles move apart and start to form spindle fibers. Does this one look different? This is metaphase. In metaphase, the centrioles are on opposite ends of the cell. You see those? Centrioles on opposite ends of the cells. What's in between them? What are all these? What are all these?
Oh, spindle fibers.
Spindle fibers. All of these lines are spindle fibers. The leftover spindle fibers, which are radiating out here, are called asters. So your spindle fibers are between the centrioles, and then we've got some asters forming too. And the most unique thing about metaphase is that your chromatids line up in the middle of the cell. Everybody see that?
Yeah.
And there is one pair per spindle fiber. And if we look at them, there's a little piece here called the centromere. God, I like this. And that centromere helps to hook that onto the spindle fiber. It attaches and holds that chromatid pair to the spindle fiber, okay? Because, you know, that cytoplasm is moving all over the place. So you'll have it doesn't look like it, but you have one pair per spindle fiber here. The cell can make like hundreds of spindle fibers, right? We still have 92 here.
So you said it has one pair of chromatids?
One chromatid pair.
Okay.
One chromatid pair, because a chromatid is actually two. One chromatid pair per spindle fiber.
Okay.
So there'd be one X on each spindle fiber. That make sense? And then you get to anaphase. And in anaphase, the chromatid pairs split. See how they're moving apart here? Ana always means apart or away. So your chromatid pairs split apart. Chromatids split apart. They're gonna gonna move toward opposite ends. Does anybody know what they're correctly called now? If your chromatid pair splits apart and one goes here and one goes here,
Chromosomes.
they're single strands now, and what are they called?
Chromosomes.
Chromosomes. So if this is a human skin cell, how many are over here?
46.
46.
46. How many over there?
46.
46 if everything went right. That's anaphase, away from the middle, apart. These slides will be on your lab exam. They will be blown up. You will have to label the phase and tell me things that are happening in the phase. Okay? Here's telophase. Telophase, we're going to start to separate into cells. Now, textbooks have gone to calling cytokinesis a separate phase, and I just don't buy it. Cytokinesis is not, I want to be careful. Do you see the word phase in cytokinesis? It's not a phase. It is simply, it happens at the end of telophase. Were you told differently?
No, I just like how you're matter of fact about it.
Huh?
I just like how you're matter of fact about it. It's not a phase.
Not a phase, otherwise the word phase would be in there.
Right.
Right? So it's a process. Now what happens in telophase, first of all, you look for the pinching in. You see the little indentation? The cell membrane starts to pinch in. Why is it pinching in?
Separate.
To separate. Okay? Your chromosomes lengthen back up to what they should be. This doesn't show it well, but your nucleus comes back. Nucleus starts to reform and your spindle fibers fade. That's all during telophase. Let me show you another slide. Put it on here. No,
There's like a,
They go back to size.
this is a good anaphase. Oh, right here.
Yeah.
Here's telophase where it's pinching in. You can't see it, but that nuclear membrane is starting to come back. And these are your spindle fibers. As long as they're still spindle fibers, people, we haven't completely divided. We're not back in interphase. So you're gonna see this slide here in your test. And hopefully you know the phases. Because all these are in different phases. Not some of them. We'll go through that in a minute. Oh, this is a good one. You see the spindle fibers here? That they're fading? Now, once that cleavage is all the way down, this is called a cleavage furrow. Once that's all the way down, the nucleus is pretty well back, then finally my cytoplasm is dividing. It's at the end of telophase. And technically the word cytokinesis means cytoplasm division. So when your cytoplasm divides, that is cytokinesis. But if I ask you the phase, it is telophase. Good? Before you leave, let's just look at these for a minute. See if you can identify the phase. So what would this one up here be?
Metaphase?
What?
Metaphase?
Metaphase is correct. See how they're in the middle? What do you think this one is? Anaphase, you're pulling apart. This one, telophase, because they're pinching in. This one,
Metaphase?
still anaphase, just pulling apart, no pinching in. This one, anaphase. This one, metaphase. This one,
Metaphase.
this one,
that's a pro.
that is prophase, that is correct. Very good. Right? We're out of time. So,
Like